Hi everyone. I'm glad to be here to give the overview talk of the placental clock dream challenge. So this dream challenge comes after two. Should I speak louder? Okay. So this dream challenge comes after two other dream challenges that we did in pregnancy research. The first two were focused on prediction of preterm birth using some information collected earlier in pregnancy, including transcriptomics and the microbiome as well. And the challenge that we are going to talk about today is the placental clock. And the goal here was to use machine learning to leverage epigenetic information to assess how the placenta tissue is aging, and from that to try to learn something about pregnancy and pregnancy complications. So the DNA methylation was proposed as a molecular readout of multiple processes that go from development to maintenance and eventually to the decline of the tissue, right? In part, these processes are reversible and lifestyle changes may affect this methylation process. So it was proposed by Hovarth and others to use methylation and to use DNA methylation to estimate what is the age of a person or of a tissue, right? You can do it with blood cells, you can do it with tissue, to other types of tissues, and so on. And the concept here is that if the methylation-based estimate of age is above what you would expect, then you will say, well, there is an accelerated aging. While if that prediction is lower, is below the expectation, then you would say, well, the biological age is less. So probably, you don't want to have an accelerated aging. You would want to have a negative acceleration if there is, if it is possible to call it that way. So previous epigenetic clocks were developed, and these range in terms of what types of data they were trained on. For instance, the Hovarth clock there is a multi-tissue clock, so it was based on information from multiple tissues, while the other two clocks at the right were derived using just blood epigenetic data. All these clocks share something in common, which is that they track very well the biological age, or at least they predict chronological age, let's say. So if you see here the accuracy of all these three models of predicting the age when you use as input epigenetic measurements in blood, all these models work well, while some other models work well in terms of predicting time to death or other, more serious outcomes. So what would be the benefit of having a placental clock, right? Why would you be interested in having a model that tells us what should be the age of these placenta tissues? So there are some big questions that we are interested in using these clocks, and one of them is, well, is this kind of accelerated aging associated with any of the obstetrical conditions or neonatal complications that those babies are going to have? Are there any particular environmental factors that influence this accelerated aging that maybe we can understand, how these factors played into accelerated aging, and then ultimately how does that affect the different obstetrical outcomes and risk in different obstetrical outcomes? And of course, ideally, well, could we use something non-invasively to make predictions of who has or who's going to have accelerated aging of those tissues? So this particular clock here by Lee et al., you're seeing here, they use the Illumina 400K type of array, and this is the prediction on a test set. And they claimed an error of about 1 week mean absolute error. So 1 week of differences between the actual gestational age, which can go up to 40 weeks or a little bit more, and the predicted values. when they used a previous clock, which was based on Illumina 27K, which was a smaller array, they concluded that the previous clock had a higher error of about 2.6 mean absolute error, which is here, right? So now this will always happen. You present your model, you show an error. If you have other people's model applied on your data, of course they're going to do worse. I mean, this is, So what we wanted to see is, well, what if we use these epic arrays from Illumina, which have even a higher coverage of the methylome, and we also put together more training data, well, and also a test data set, a large test data set. Can we develop a better clock, and what can we learn out of that, right? So that was the goal of this challenge. So we formulated these as a DREAM challenge for the advantages that by now DREAM people that participate in DREAM are aware of. From some point of view, we organize this challenge in the most possible strict way, and these are the three key features. So this was a model to data challenge, meaning participants could not see the test data, any information about the test data. So no feature values, of course, no outcome, because they were blinded to that. Participants could submit models and see their accuracy on a leaderboard set, but that was not the way we judged the models. The models were judged on a test set, so they did not even have access to accuracy, to the actual accuracy of their models. And then finally, the other component of this, which made it a particularly challenging task, was really the size, because on most, if you take most people that do modeling, if you give them 1 million rows and 2,000 columns and tell them to make models, they are not going to like the setup, right? So they will need a cluster, they will need some bigger resources. So this is a training set, 1,742 samples. We collected this from the GEO. In particularly, we want to highlight that we did find one hidden gem in the GEO. So this third largest training data set was having the methylation data, but the the gestational age values were not there. So we did obtain them from the investigators, and we could enlarge this training data set. Now, just to give an idea about how challenging this challenge was, I'm going to show you the distributions of what we are trying to predict, which are the gestational ages. So some data sets only have early gestational values, which are here. Some other ones have only term gestation ages. So it's not that ideal setup where you have all outcomes in each study so that you can eventually do some bias adjustments. So the second you are trying to adjust to do a batch adjustment, you are removing signal automatically. So, good luck with that. So this is how we organized the challenge. So first of all, we split it in two sub-challenges. In sub-challenge 1, the goal was to try to build the clock only for 450,000 CPGs. These 450 CPGs were present in all the training samples. And this is a distribution here of the gestational age values. And then this is the data set that we have generated. So again, this was the easier of the two sub-challenges because the size was only 450K, and then they were not missing anywhere. So the participants would submit a Docker that describes completely what system requirements are, how do you what do you need to apply that model to a new data set, right? So this is using Docker technology. For the sub-challenge number 2, we provided the 850K profiles for all the samples that we had that information. For the ones that were missing, we provided an imputation, but we told participants, hey, you should use yours or maybe not use it. And we will see that actually, this imputation was a possible issue here. And I also want to highlight that, as all the DREAM challenges, there will be no challenge without the test data set. So the test data set was generated at Wayne State University with support from the perinatology research branch, which was an intramural unit of the NIH housed at Wayne State. So the epigenetic profiling was done at the University of Michigan using the EPIC arrays. Now, regarding the test set had a multitude of possible phenotypes, meaning we had different disease groups such as preeclampsia, both early in pregnancy or late. We had women that delivered spontaneously preterm, and we also had the majority, normal pregnancies. So we had this full spectrum of samples that were included in the test set. This is a principal component plot just to show that the test data, which is here in the middle, falls somewhere in between all possible, epigenetic profiles out there. But again, we did not do any particular normalization to this. We let participants to decide what do they want to, use. So the challenge ran from June to beginning of September, and we had 74 participants and 49 submissions, and 7 separate teams were able to make a successful submission that we were able to score. So here are the results on the leaderboard. So the leaderboard had 100 samples, part of the original studies that were included in the training, right? So they were separate, but they came from the same study. So based on this, we had the team OPPEC that was number 1 with the root mean square error of 1 unit, one gestational age week. However, as I was saying, this is not how we judge the teams in the end. We use a separate test set. And as you see, when you use a test set, which is independent from the leaderboard, things can go in both directions. You can go up or you can go down. Of interest, the team that we ranked in the end as top in this challenge, root mean square error of 1.24, had pretty much the same accuracy on 1.28. I mean, on the leaderboard set, it was pretty much the same accuracy. So actually, we were really happy to see that. So these are the 3 top teams. So we had for team number 1, Heriandi, Surfiana, and Emily Su. And then we had for team number 2, Tushar Patel, Elena von Bommel, and Stephen Hoffman. And team number 3, Ibrahim Al-Sagaf and Chen Wan. And you can see the affiliations. I would want to just make a general comment here is that of interest, right? The top team had expertise in obstetrics and computation. The second team had expertise in aging in general and computation. And then the team number 3 had only computational background from what I could glance looking at their work. So, it was interesting to see that domain-specific knowledge somehow makes its way and makes this, allows for better models to be found. As in all challenges, we want to know whether the test set that we provided allowed the opportunity to rank the teams in a way which is robust to possible, fluctuations in the test set. So if we did a bootstrap by taking bootstrap samples of the test set and we would do the ranking over and over, actually we would get these distributions of root mean square errors that you see them here on the y-axis. And actually the top 3 teams could be, ranked in a way which is unambiguous in this situation. Now, this is how the predicted gestational age looks like versus actual gestational age by the team number 2 model. And you do see here, the different colors representing the different phenotypes of those placentas. But you can appreciate that, the model is reasonably well calibrated, meaning that the parity line passes pretty much through the cloud of points. And that was even more the case for the team number 2. And we are also, very happy to see that. And if we do a wisdom of crowds, not a perfect one, but we will just average the top 3 teams, we will get actually better accuracy. Might be slightly biased because, again, we rank them based on the test set, but still, it suggests that there is more there to be learned out of that data. And it's just that people run out of time and run out of memory trying to fit the different models. Now, there is always a question, when you organize a dream challenge, did you have enough skill involved in those, in those models that were submitted? Did people do, reasonably well? Could you have done much better if you try something else? And to answer this, at least with the information that we have so far, we did use what is called AutoML. It's a package, AutoGluon is actually called, so it's automatic machine learning. It's a package created by Amazon with the idea that would give people that don't have necessarily a background in machine learning, would give them just 2 lines of code to fit a model and then apply it to a new test set, right? So we really like that. So the way it works, so it's implemented in Python, and then the way it works is it tests and tunes many different types of models, then it stacks those models and gets a final prediction. So this is just briefly, but it's a lot of thought that was put into this. So in this case, it was applied to pretty much tabular data because this is what we have, but AutoGluon is out there and it can be applied to other types of data, multimodal data, and so on. So why would we choose AutoGluon as opposed to other? Well, this is a PhD thesis from the Netherlands, and they compared AutoGluon with other automatic machine learning pipelines, and they found that is, based on their 104 data sets that include regression, classification, and multi-class prediction problems, AutoGluon would always come first based on 4 hours of computation time allocated to a given task. So now, well, it didn't necessarily work as advertised in the sense that, okay, so you start your cluster, you say, well, you have 2 days to finish this, and by the end of the 2 days, your process is dead because, the university shuts down your session, but your model didn't fit because it actually needed 4 days, right? So in the end, what I'm showing you here was a model obtained after 24 hours of allowed computation time, but actually 40 hours passed since it was started. Luckily, it was right before 48. I would have it killed again. So in the end, this AutoGluon would have been somewhere here in between team number 4 and team number 5. So that gives us a sense that the top models that we got were actually very good. Now, how are these top models that we got different from what is out there already, compared to the other placental clock? So yeah, if we look at the accuracy, mean, absolute error, yeah, we're doing a little bit better. But of course, this is the data set we use to rank these teams and to choose those models, right? So I'm not necessarily impressed by the 0.99 here compared to the robust placenta clock, which was proposed by Howard et al. So we're doing a little bit better, but that's not the thing that is going to impress you. I think the first thing to notice, looking at these existing 3 clocks, the first one is the robust placenta clock, which uses both normal and complicated pregnancies in creating the model. The second one is the control placenta clock, which tries to use only as normal pregnancies as possible, right? And then the third one is a refined placental clock. Now, if you look at all those 3, you will notice the following, that all the early samples are over-predicted, right? So the predictions here and here and here, they are all above parity, right? So what does that mean? That pretty much means that there is accelerated aging in those placentas. This is what it is saying, right? Now, if we're breaking down those predictions by the different phenotypes of preterm birth and term birth, we would see that this accelerated aging appears to be present regardless that women were induced to delivery because they had preeclampsia or because they delivered spontaneously with rupture of membranes here, PPROM, or after spontaneous labor with intact membrane. So what it seems to happen is that if you use the previous clocks, we would have accelerated aging in all these preterm groups, which we don't think is going to make sense because, for instance, a study from the perinatology research branch that looked into this particular group, which is the spontaneous preterm labor, found that there is going to be a fraction of cases with accelerated maturation or with decelerate, so delayed maturation and accelerated. So you have a fraction, and those two fractions should be at the ends of the spectrum, would not justify to shift all the mean predictions, right? So if we use the top DREAM model for this case, instead of having about 2 weeks of acceleration, we only have about half of a week of acceleration, right? So there is still a slight trend there, but we still think it's an artifact, and it is really not people were thinking. So what are the implications of these new models? So there was a study that was claiming that there is accelerated placenta aging in early preeclampsia, okay? And that was done by comparing the methylation-based age with the chronological age, which is pretty much what I have shown you there. And then this was happening in early preeclampsia, but this was not happening in late preeclampsia. So what we believe that is happening is just because these models are trained with mostly term samples, are going to predict better and with less bias the term samples, but they will always over-predict the preterm samples. So when we take that main et al. Clock and we apply it to the entire training data set that we have, you will see that all of these samples will be over-predicted. So then, just to conclude here, we believe that the models that were found in this DREAM challenge are not only slightly more accurate, but they're importantly less biased than the previous models. And we think that this has implications in the way people are going to evaluate the relation between accelerated aging and obstetrical disease. And well, compared to the automatic machine learning, it looks like the skill and the insight into the domains of the different teams allowed us to find better models. And with this, I would like to acknowledge members in my lab, especially Gaurav Bhatti, who is a postdoc. He did a PhD with me at Wayne State, and he was a main driver here of this challenge in all these aspects. And also, I had lots of volunteers that helped with setting up the challenge or testing the submission system to see that it works. And of course, we have the co-organizers that I would like to acknowledge here. A big thank you to Dr. Roberto Romero, who really, allowed for these studies to be performed and collect these samples and generate the data. So with that being said, I'd like to thank you and invite the top team to present their approach.